Hello, my name is Mrs. Tyler, and today we're going to be doing some biology revision in the form of making a knowledge organiser. A knowledge organiser is a short document where you try to reduce all the content from maybe 3 or 4 lessons down to a single sheet. Today's one is going to look a bit like this. We're going to be looking at fighting disease, so we're going to be looking at the biological response of the immune system, but we're also going to be looking at the role of drugs and drug development. So there's a lot of information here. Please make sure that you do pause the video if you get behind. If you're doing this in a class, ask your teacher. I'm sure they'll pause it for you. If you're doing it at home, I'm absolutely delighted. Well done. So all you need is a sheet of paper and landscape and a pen. Let's get started. So, as always, we're going to start with our title. I'm going to try and draw it in the middle. I usually don't quite manage it. And today we are looking at fighting disease. It's important that the paper is in landscape so that we can fit everything on. And as usual, we're going to divide our paper up so we can keep track of where we are and get it all on one page. So we're going to divide it down the middle into 2 halves. And then today's topic, we're going to do about the immune system up here. Now that's quite a big topic, so I'm going to need quite a lot of space for that. So immune system. So I'm going to use about 3/4 of the page, so about the bottom quarter. It's going to be about vaccinations. Draw boxes round so we can see them nice and easily when it gets all filled up. And on this half, annoyingly, it's going to be the other way around. I'm going to separate it into about 1/3 for information on drugs. And then the bottom 2/3 is going to be about drug development. Let's start with then the immune system. So before pathogens can get in, they've got to get past barriers. So I'm going to draw a little gingerbread man here. Don't draw him too big because I'm not that bothered about him. Here we've got our little man. And we've got natural barriers that stop pathogens, microorganisms, from getting into our cells. The most important barrier covers the entirety of your outside, and that is your skin. So that's waterproof. It stops pathogens getting in. Unfortunately, occasionally you'll cut or damage your skin and then the pathogens can get in. You've also got systems to stop pathogens getting in through different orifices. So if this is his face, that's his nose. You've also got hair and mucus to trap pathogens that might try and get into your respiratory system. And you've got things like hydrochloric acid in your stomach. So you've got barriers to stop the pathogens from getting in. But if they do get through these barriers, so you've got a hole in the skin, or they don't get trapped in the mucus, or they're not killed by the acid, then we need an extra defense system. And that's our immune system. So when the pathogens get in, we need our immune system. And our immune system runs on a type of blood cell called the white blood cell. going to shorten white blood cell to WBC because we are going to be writing it a lot. White blood cells then, as the name suggests, are found in the blood, and there are loads of different types of white blood cells. Some of them have got funny shaped nucleuses, so they're like lobed like that. Others have got massive nuclei, and they do a variety of different jobs. But the 3 jobs that you need to know about at GCSE are as follows. The first job that some white blood cells can do is called phagocytosis. And it means to engulf and digest bacteria or pathogens. Phage means to eat. So it literally means cell eating. Phagocytosis then happens in quite a few different types of white blood cell. And what they do is they detect your pathogen and then they send out their cytoplasm, and it makes like little arms that wrap around the pathogen. So I'm going to draw my white blood cell. Here it is with its funny shaped nucleus. And here is our bacterium. Okay. So it's wrapping its cytoplasm around it. The name for this type of cell that can wrap cytoplasm around pathogens and engulf and digest them is phagocyte or phagocyte. And again, we've got phage there to eat. So they are eating cells. And so it sends out these arms, they're called pseudopodia actually, pretend arms. And they eventually seal the pathogen inside. So here's our white blood cell again. Here's our funny shaped nucleus. Now we've got our bacteria trapped inside the phagocyte. And the phagocyte squirts enzymes in. And those enzymes chop up the bacteria and kill it. And essentially, the phagocyte then takes on board the protein from the bacteria and uses it for its own means. So the phagocyte secretes enzymes that digest the bacteria. So your blood has got phagocytes in it, and they live particularly in places like your lungs. You've got cells called macrophage, which is a type of phagocyte, and they're patrolling around looking for any pesky bacteria that get in, and they engulf them and then digest them and kill them. Another type of white blood cell then can produce chemicals called antitoxins. I'll just underline that as well. So here we've got a white blood cell again, a blob-shaped cell. I'm going to do this one with a big round nucleus. And some types of bacteria secrete poisons. So here's my bacteria, and here's the poison it's secreting. And the poisons that it secrete are called toxins. So that's a bacterial toxin. It's the toxin that causes the symptoms of some diseases. For example, in some types of food poisoning, the bacteria squirts out these poisons, and it's not the bacteria itself, it's the toxins that it makes that gives you diarrhea. So we've got cells that can secrete molecules that attach onto the toxins, and we say they are neutralized. We stop them from working. So that would be an antitoxin there. So I'm just going to put the toxin often causes the symptoms. And the last one is the most important one, and that is that some types of white blood cell can make antibodies. So in your immune system, then, we've got lots of different types of white blood cell. Some of them are called B cells, and they can make antibodies. And they are made with a massive variety of different shaped receptors on them. I'm going to just draw 3 here. So here's 3 B cells. Put the nucleus in. And basically, they've got different shaped receptors. So here we've got a curvy one. Here we've got a triangular one. Here we've got a square one. And these receptors are going to match up or be complementary to different shaped antigens. An antigen is a protein on the outside of a pathogen. So I'm going to draw here my pathogen, drawing a bacterium again. And this one has got spikes, triangular shaped antigens. So antigens are usually proteins, but they don't have to be, found on the outside of all cells, and they're different to yours. All right, so you might have circular shaped antigens, and this bacteria's got a different shaped one. We say they are non-self. So it tells white blood cells that is non-self. So when these white blood cells come into contact with an antigen, you can see that this pointy one is going to fit into here. I'm just going to draw it there. And when that fits into there, it activates this one. And this one has now been selected. And instead of being just 1 or 2 of them, you will make thousands of them. So now I'm not going to draw thousands, I'm going to draw 4. All of them with the same shaped receptor. But here's the clever part. This receptor can be fired out into the blood as an antibody. So these cells can release billions, trillions of antibodies. Antibodies are proteins that will stick to antigens. So here's our antigens. And now this poor bacterium is going to get antibodies stuck to it. So when it's activated, it divides or clones and makes lots of copies of itself, and then can fire out millions of antibodies, and they stick to any antigens. So wherever that pathogen has got in, wherever the infection is, antibodies can get there. So we're going to say white blood cells. Oh, I could have shortened it to WBC. Make millions, billions, trillions of antibodies. And the antibodies attach and kill the pathogen. So antibodies are our best form of defence because once we've made them, they can travel anywhere in the body. Right. And they usually will kill a pathogen within a couple of days of being stimulated. The antibody level will be so high that all the pathogens are killed. Now, we can't keep making white blood cells and we can't keep making billions of antibodies. It uses up far too much energy. All right? It's one of the reasons why we feel tired. So instead, we also make memory cells. I'm going to draw one here. So they look like these cells that are making antibodies, but they are very long living. I'm going to give it a brain. I'm going to give it a little smiley face because these are really useful cells. And memory cells stay alive, ready for any other infection. If this type of bacteria comes back in, we've got a few of these ready to go. Instead of just 1 or 2 cells, we've got several hundred of them ready to go, and they make antibodies so quickly that they kill the pathogen before you even get any symptoms. So they're ready to quickly make antibodies in future infections. So the immune system then is based on white blood cells in the blood, engulfing and digesting pathogens, releasing antitoxins to neutralize toxins or poisons, and importantly, make antibodies. And those antibodies stick to and kill the pathogen. Vaccinations then are a way we use our own immune system to protect ourselves from future infections. So a vaccination is when we inject, although sometimes it can be eaten or squirted up the nose, small amounts of dead or weakened pathogens. So instead of a live bacterium, you will have a dead one. So here's our bacterium, here's its antigens, but it's dead. Okay. We can even sometimes just inject the antigens. So sometimes we can make the antigens and inject those. So when a dead bacterium is injected, your white blood cells don't know it's dead. And so they go through the process of selecting a white blood cell, activating it, making antibodies, trying to kill the pathogen, but it's already dead. But importantly, it will also make memory cells. And these memory cells are going to protect you, depending on the vaccine, for a very long time, sometimes less length of time, but certainly give you an added protection. So white blood, I've written it again instead of doing WBC, you could just do WBC. White blood cells make memory cells. And that means if you get the real pathogen, antibodies are fired out so quickly, right, because this can clone us that much quicker, and there's lots of them, that it will happen before you even get symptoms, before you even know you're ill. And that is when you're immune. So if you get the live pathogen, so the actual disease, antibodies are made much quicker. And that is when you're immune. Or immunity. So when you've got immunity, it's not that you can't get the bacteria. You get the bacteria like everyone else, but your memory cells make antibodies much quicker than everyone else and kill it pretty fast. So these are all biological ways we protect ourselves, but also we can use chemicals to help protect ourselves or to relieve symptoms. So drugs are chemicals used to cure or relieve symptoms. And they often came from natural sources or originated from natural sources. Originated. So the examples you need to know for the AQA specification are willow, which is a type of tree. The willow bark originally contains the compound that is now known as aspirin. Aspirin is a painkiller. Another type of plant that was used were foxgloves. They're long strands of purple flowers. They grow quite tall. And their leaves, and the flowers actually, contain different forms of a drug called digitalis. Digitalis. And digitalis is used to treat heart problems. And lastly, mold or fungus was originally used as the original source of antibiotics. Still is today, actually. And antibiotics, remember, kill bacterial infections. Not viral. I want to quickly mention then some of the problems we're having with antibiotics. You do about the effect of antibiotic resistance in much more detail in Unit 6, which sits in Paper 2. But I'm just going to mention it here because the problem with antibiotics is that some bacteria are becoming resistant. So I'm just going to put the subheading antibiotic resistance. So antibiotic resistance is when bacteria mutate. So their DNA changes and it's like they get a superpower. They mutate and it means that antibiotics won't kill it anymore. So antibiotic resistance means a bacteria has mutated and the antibiotics won't kill it anymore. So sometimes it's the cell wall, sometimes they kick the antibiotic out. However it's mutated, it can't get killed. However, all the other bacteria that aren't mutated will get killed. All the other bacteria are killed. And this is bad news because the resistant ones aren't. And so they survive and reproduce. And so instead of one mutant bacteria, you could have hundreds of thousands or millions, and they're not killed by antibiotics, and they make toxins and can make you ill. The resistant one survives and reproduces. So this really is an example of evolution. We also saw it in the COVID epidemic, actually, whereby the pathogen mutates, although COVID was a virus. I'll just start again, so I'll go back to here. So one survives and reproduces. So this is an example of evolution, where one individual is selected over the other and survives and goes on to reproduce. So MRSA is a type of bacteria that has mutated. Antibiotics have stopped working, so it's antibiotic resistant. And it's now called a superbug. It can survive where others are killed. On to our last big section then, and we're talking really, when it says drug development, we're talking about drug testing. So when we find a substance and we think it might be useful, then we need to make sure that it's safe. And we do that in 4 stages. The first 2 stages are called preclinical trials. The second 2 stages are called clinical trials. Let's start then with stage 1. And in stage 1, we're not using an organism, an animal, or a human. We're using just human cells or tissues. So I'm going to draw a petri dish as my oval. Let's draw some lines down like that. And inside my petri dish, I'm going to draw some cells. Obviously, you wouldn't be able to see them because cells are microscopic. I'm going to just draw the liquid they're grown in. So to grow human cells in a jar is quite tricky, actually. So you need growth medium to keep it alive. So that's just a liquid full of glucose. You also need to make sure there's plenty of oxygen around and buffers so it doesn't get too acidic. And the cells themselves, here's a weird one, are often cancer cells because you need them to keep growing. You need them to survive. So you need your human cells, whatever it is you're interested in. So we get some cells either from a patient or from some cancer cells that we buy in. We put them in a petri dish and then we add the drug. And then you can just simply use a microscope to look at the cells, see if they've been killed or not, see if they've changed. So add the drug and then use a microscope. To see the effects. So this is really clever. All right. The problem is that you've only got one type of cell. So if you're wanting to test a drug that you think might affect brain cells, you'll have neurons in here, but you don't know whether it affects kidney cells or muscle cells. So you can't see the effect on other cell types. Problem. So although it's a good starter, we can't use it straight away into humans because we've no idea, although it's all right in brain cells, is it actually going to give somebody a heart attack? So we go to stage 2, which is animal testing. And the reason we use animal testing in the UK is because of this, because we can't see systematic problems. Okay. And so with animals, we are testing for efficacy. Efficacy is the word you need to know to say whether the drug works. And we're testing for dosage. See how much should be given and see whether it's toxic. Also looking at toxicity. Now, because both human cells in a jar and animal testing aren't on humans, we call it preclinical. So both of these are preclinical drug trials. Human cells first, then on animals. If we've tested on 2 different types of animal and we found it safe and we think it's going to work, then we will go into clinical trials. So this is our 3rd step. And the first type of clinical trials is on healthy volunteers. So these are individuals, they're often paid to take the drug and they're checking to see for side effects. So it might be, for example, that the drug gives you a terrible headache, but a mouse can't tell you that it's got a terrible headache. So you need to put it into humans first to see what those side effects might be. If it passes phase 1 clinical trials in healthy volunteers, then it will go on to the next stage of clinical trials, phase 2, which is on patients with the disease. Here we're testing for efficacy again. Does it actually work? And we're looking at optimum dosage. Now, both phase 1 clinical trials on healthy volunteers and phase 2 clinical trials on patients often use something called a placebo. And a placebo is a fake drug. All right. So it needs to be exactly the same as the real drug. So if the real drug's an injection, then the placebo needs to be an injection. If the real drug's a pink tablet, then the placebo needs to be a pink tablet. But it's fake. It's got no active ingredient. So it's just a sugar pill. And when you give somebody a placebo, there's a demonstrated and recorded effect called the placebo effect. And this is when you give someone the fake drug and they think they feel better. So the placebo effect. When you give someone this fake drug, and they feel better just because they think they should, because they've had a tablet. So to get round this then, drug trials, both on healthy volunteers and with patients with the disease, are often blind trials. So you give half of the volunteers a placebo, the other half the active drug, and you look to see if there's a difference between them. So blind trials are when the patients don't know if they've got a placebo or the real drug. And another test they can do is called a double-blind trial, which is when the patient doesn't know, but equally important, the medical staff that are doing the trial don't know either. So neither patient or medical staff. And that's really important because they found when they started doing blind trials, sometimes the people handing out the tablets would be extra nice to the people that were getting the real drug and extra interested compared to the people who were getting the placebo, because they knew that the placebo people shouldn't really be changing how they feel. So then they had to invent double-blind trials to stop doctors and nurses showing bias, being extra nice to some people compared to others. And there we've got it. As always then, well done. There's a lot on there, isn't there? And now your job is to go away and use read, cover, recite to learn this content and then test yourself. So, for example, if I wanted to remember phagocytosis is engulf and digest. Phagocytosis is engulf and digest. Cover it up and say it to myself out loud or just quietly in my head. Phagocytosis is where they engulf and digest. So I'm going to go through, rereading it, covering it up, saying it back to myself, and then I'm going to give myself a test with a few questions. Well done.